Good afternoon, everybody. And it is indeed a great pleasure to have you all on board today for these two presentations. One by Associate Professor Archana Baulaksimo from CBBR, and the second one is from Dr. Mohamed Ali from the University of Mauritius, Department of Health Sciences, if I'm not mistaken. So indeed, as Madhvi just rightly pointed out, we had 26 projects approved under that particular special call, which was launched last year in the confinement period. And 13 of them have already been completed and disseminated, including the two that are going to be disseminated today. As I said earlier, this is a special call. We have, at the level of the council, launched a various numbers of special calls to be able to respond to the needs of the country with regards to research, development, and innovation. So just after the COVID-19, we had the Wakashu special call as well. And this year, we had a number of calls regarding fast-track innovation initiative, enterprise innovation booster scheme, public sector transformation scheme. And the MRIC is moving ahead with other innovation initiatives as well. We are launching our national campaign for innovation.
We cannot hear.
Professor, I think you are mute.
Yeah.
I'm very sorry about that. So very good afternoon, everybody. Just I wanted to highlight the fact that Madhvi highlighted that we had 26 projects approved under that particular project, special call, and 13 have been completed, including these two which are being presented today. The MRIC has been, after the special call for COVID-19 in March, April last year, initiated several other calls as well, including the Wakashu special call, the Blue Resilience, the Fast Track Innovation Initiatives, Public Sector Transformation Scheme, Enterprise Innovation Booster Scheme, and in the coming weeks, we are launching a national campaign for innovation, which will culminate in the organization of Assises de la Recherche et de Développement et de l'Innovation, which would be at the end of this year or most probably in January next year. So as you see, we are targeting innovation with regards to not only the upper part of the pyramid with industries, academias, and SMEs and incubators. We are also targeting grassroots innovation with regards to our campaign, and also trying at the level of the Assises to chart the roadmap for innovation for Mauritius. As you may also have seen at the last Cabinet of Minister meeting on Friday last, MRIC is opting to change its mandate to move towards directing research and innovation and being the apex institution of a country. So with regards to, indeed, with the help of all stakeholders, I'm sure that this vision will be materialized and the mandate of the MRIC will be reinforced accordingly. So today we have two presentations emanating from the COVID-19 special call. One is from Dr. Archana Baulaksimo from CBBR, who has developed a special nanotech mask for viral protection. And the second one is from Dr. Mohamed Ali, who will be talking about the potential of our floral biodiversity to combat COVID-19. So without much ado, I'll leave the floor to the presenters for their outcomes in terms of their projects, and also to take afterwards question and answers. Thank you.
Thank you, Professor. We'll now move to the first presentation, which is the Nanotech Mask for viral protection, which is going to be made by Dr. Archana Baulaksimo and Mr. Kendall Tang. Dr. Bao Laximan is the leader of the Center for CBBR, the University of Mauritius, and her focus is on the engineering of biomaterials-based nano devices. Mr. Tang Drieski is the CEO of RT Knits Ltd and is also an electrical engineer by training. Before we start, ladies and gentlemen, I will invite you to put all your questions in the chat box and address it to the MRIC administrator. We will put, you are kindly requested to put the name of the presenter to which the question is being addressed as well. And we will take all the questions at the end. So I will now invite Archana to start her presentation. Archana, the screen is yours.
Thank you, Madhvi. Let me put this on share screen. Before I start, I would like to thank all those that have tuned in today for the webinar, and a special thanks to Dr. Victor. Victor is from the United Nations Economic Commission for Africa, and he's present today. And the UN ECA is funding the next phase of the project. Thank you, Victor, because I saw you on the list of those that have logged in. And today what we are going to try to give to you an overview of how we made the Nanotech Mask. And this Nanotech Mask is a collaboration between industry and academia and university. And we had a number of challenges that we had to look at before having the final prototype. And you might ask yourself, why do we still need an antiviral protection? And we do need antiviral protection. If you look at what is going on nowadays, if you look at the latest data, this is the 27th May data from the Johns Hopkins COVID-19 Resource Center, you can see that the number of cases is increasing, the number of vaccine doses is also increasing. But if you look at the vaccination coverage around the world, we are still around 10 to 20 doses per 100%. And most importantly, are the variants. If you look at the distribution of variants, this is a site that collects all the genomic data concerning variants, where all countries are sending their data. You can see that there's a good distribution of variants around the world, and Mauritius has got the 20AB clade variant. And the latest one, 2 days ago, Vietnam talked about having the new hybrid variant coming from the UK and Indian strain. And, I found yesterday a very simple definition of variants, because many people, they talk about variants, but they don't know really how these variants are manufactured in the host. And these viruses, the variants, they come from what we call reassortment. That is, when the virus is in a host, it sees another virus or it sees the DNA material of the host, they start making new copies of the gene, and they mix up all these copies of genes, and this is where the new viruses are assembled and formed. This is a simple definition how variants are formed. Now, why is it important for Mauritius? We have seen what is going on around the world. Now, we have been seeing emergence of clusters around the island, and having a viral protection is very important because it will allow us to mitigate the formation of these clusters. And we can also reduce the widespread distribution in the community, because we can see cases coming up in small villages in the north, south, east, everywhere. And if we do not have the proper viral protection, our daily activities and well-being will be impacted with all these cluster formation. And very importantly, we have to protect our healthcare workforce with the recent events that we have seen concerning our healthcare workforce and COVID-19 infection. I think it's very important that we protect this healthcare workforce. Now, it has been predicted that the COVID may become like a seasonal flu, and many researchers have said that eradication of that virus is basically impossible. and we should expect regular outbreaks. Therefore, coupling vaccination with protection is extremely important at this moment in time. And to come back to how come we got the idea of making the Nanotech Mask, at the Center for Biomedical Biomaterials Research, particularly in the Biomaterials Drug Delivery and Nanotechnology Unit, we have three main focus. That is, we look at building the country's research intellectual capital by training PhDs and postdocs. And then we have our research and innovation thrust, which is mainly on the design and engineering of smart materials. And we look basically at locally available resources because we need to be independent in our process. And also we look at high-end analytical services to industry, particularly at the assistance to industry in uptake of nanotechnology. All this, we do it by taking into consideration the SDGs, which is extremely important. Therefore, we've been working on tissue regeneration scaffolds for the past 10 years. And we can make multiple architecture to mimic the tissue. What is a scaffold? For those that have never come across this word, a scaffold is meant as a support for growth. Therefore, a scaffold in the body will also mean it's a support for the tissue to regrow. And as we are in this field, we can make different types of architecture. You can see nanofiber scaffold. We can have a 3D scaffold, different types of these scaffolds. And using this scaffold expertise, we have plugged this in the Nanotech Mask. Kendall, now I will let you continue on RT Knits.
Good afternoon, everybody. So, in fact, for RT Knits, it's the first time that we are working on a project together with the CBBR. We are very pleased to have taken this decision, and so far our collaboration has been very successful. So who is RT Knits? So we are a fabric and garment manufacturer. We have been established since 1970. We were the first textile company. In fact, it was even created before the EPZ sector. We are currently exporting to Europe, USA, South Africa, and regionally also. We are currently employing about 1,900 people. Last year, we were the first textile company to sign what we call the Science-Based Target Initiative to commit to carbon neutrality before 2050. So we are currently working on a big project to become carbon neutral. Archana, can you move the slide? Thank you. So we started with looking at what currently exists. We all know that surgical masks exist, and then there are the respirators, the N95, N99. They are costly, they are non-washable, and they are not made locally until recently, I think. There's a company who started to manufacture surgical masks, I think, last week. And so our starting point was to look at what we could improve on these masks. Surgical masks are very cheap to manufacture. They can be manufactured in a very automated way, but they are most of the time single use. They rely on what we call electrostatic properties to block the particles. But when we wash the mask, these electrostatic properties reduces. But what we call the melt-blown layer found inside these masks are still very fine to compare to most textile fabric. So the filtering capacity, filtering efficiency of these masks are still superior to most, I would say, textile-based fabric mask. And one disadvantage of these surgical masks is that they don't seal very well around the face. So even though that they have a good filtering efficiency, the fact that they don't have a very good sealing around the face makes them problematic. And then we have the respirator category. The respirator category sometimes can be formed by up to 5 layers. There are different layers. The most important layer is the melt-blown polypropylene. Most of the time they are quite uncomfortable, less breathable, because the membrane layers are quite dense and have a resistance on breathing. And so these are engineered to block, obviously, the virus, which is between 50 nanometer to 140 nanometer. But of course, we are not blocking the virus directly. We are blocking the droplets, the aerosolized droplets which contains the virus. So the tests are not done on 50 nanometer or 140 nanometer droplets. They are done on bigger droplets than that, which are created when we sneeze or when we cough. So CBBR developed a nanofiber scaffold, the nanofiber, which is a kind of membrane that is added to the textile mask that we made. Can you move to the next slide, please? So we applied, Archana, can you move to the next slide?
I'm changing the slide, but it's not moving. Maybe the host can...
Try to help you with this one. Yogesh, can you?
No.
Try to stop sharing and then...
There you are. There you are.
Yeah, so we applied the design thinking methodology, start with empathizing and then defining, ideate, prototyping, and testing. So it's a process that helps us build a better product, starting by researching our users' needs, and then state the users' needs precisely and define the problem, and then through a brainstorming session, identify other ideas that can solve this problem, and create a solution, and then test it and improve it until we reach a satisfactory product, or the product that we want to make. So with the Nanotech Mask, our requirements was to have the best level of comfort possible with a good breathability, because if comfort and breathability are not good enough, people won't wear it or they would remove it constantly. As I explained, the seal around the face is very important, because we can have material that can filter the virus, but if the seal is not good, then there will be leakage, thus jeopardizing the filtering efficiency. It also needs to have capacity to retain sneezing. In fact, one of our goals of making this Nanotech Mask is also to have it working both ways, to retain sneezing particles, but also blocking particles that may be found in an aerosolized form in the air. And it also needs to be washable and durable. So in order to have very comfortable mask and also a low breathing resistance, we needed to design a mask which has a large breathing area. And the design of our mask is quite unique in the way that we've measured the area. It is one of the biggest available on the market, because when the material is sticking to the face, this area is not used, because it cannot be used to when we are inhaling, when we are exhaling and inhaling, that area can't be used. So it is only a material that is not sticking to the face that is usable. So we created a mask that has a big breathing area with hydrophilic material inside, so as to absorb, for example, particles that may be generated during sneezing or coughing, and also hydrophobic outside, because the mask need to be tested for splashing resistance. So hydrophobic will help in passing this test. The soft fabric gives it comfortable, it made it comfortable for the user. and the fabric needs to be durable because we want to make the mask reusable. So we don't want it to look old or used after several washes. So we have chosen some specific fabric to make it more durable. We work on the face, on the fitting, so that to make sure that there's no leakage around the mask. And of course, we included the nanomembrane that was made together with by the CBBR, and then we tested it. So you can have on the left-hand side an idea about how the mask is. In fact, nearly only the edge is touching the face. Nearly the whole fabric area is not against the mouth or the nose. So you can see on the right hand side the different layers. So outside is the black durable hydrophobic layer, and the inside is a hydrophilic material, and in between we have this nanomembrane. In fact, there are other layers too, but these are other fabric layers. When we increase, in fact, the path, it improves the filtering efficiency. So having more layers, in fact, improves the filtering efficiency. And then I think we went to the tests. So these are the tests done by Nelson Labs. The nanomembrane material has itself been certified at 99.95%, so nearly blocking everything. So this was a viral filtration efficiency test done with a virus that is even, in fact, smaller than the COVID-19. So this 175X174 bacteriophage is about 30 nanometer, whereas the COVID-19 is between 50 to 140 nanometer. But again, as I said, we don't test directly with these viruses. These viruses are contained in droplets that are bigger particles. And then the mask also passed all the breathing resistance, splash resistance tests as required. Archana, you want to add something on these test reports?
Yes, because on the viral filtration efficiency test, I mean, in the aerosols, in the droplets that we use, there were millions of virus loaded in that. And also, we did the test at increased challenge level, which means that the velocity of impact was increased by 16 times, I think. They increased it by 16 times than the normal velocity impact that the virus would have when you're wearing a mask, which means that this is an increased challenge level viral filtration efficiency test. We chose that on purpose just to see what is the maximum that we can go. And also the test was done after washing, and we had similar results. And we, again, have to wait. Okay, therefore, what is the status of the Nanotech Mask? In December 2020, we got the UN ECA Innovation Award. And in April 2021, we acquired a semi-industrial pilot nanospinner for upscaling, because once we have the prototype, we should be able, it's not 5 nanomembrane per day that is going to allow us to look into upscaling the process. Therefore, we acquired the equipment in April 2021. And then in May 2021, we got the first roll of nanomembrane. You can see the picture here. Therefore, we can make meters of nanomembrane. Therefore, we have increased our production capacity already from 10 masks per day to 1,000 masks per day. And we can also increase that. It depends on the speed that we can roll out the nanomembrane. And this next step is being funded by UNICA. and the upscaling of the production of the nanomembrane. That is, the Innovation Award was obtained so that we could upscale the production. And we have a short video which we want to show to you. And we have to wait for the screen to change. Here, we want to show to you a short video of the making of the Nanotech mask, and you would understand that we can't get into all the technical details because there is IP involved, but this is a short video of how we made it. So this was the short making of the Nanotech mask. And what is the future of these nanomembranes? That is, once we've got the prototype for the antiviral filtration membrane, we want to do more. That is, we want to have these smart technologies for social impact. And nanomembrane has got application in health, energy, and water. And we want to look at fuel cells to improve fuel cells. We want to look at water filtration and then air filtration. And I have to say that we have got a very good teams that is both from here and from RT Knits. These two teams, they are passionate for innovation, and we believe in uplifting the research and innovation landscape of Mauritius. And we, more importantly, want to create opportunities for this highly technical workforce. And this project is in line with the industry university collaborative efforts for social impact. And I can't, neither me nor Kendall can't finish this without asking you to wear your mask. We need to couple vaccination and protection. This is the first generation only of Nanotech Mask, where we have redeployed our expertise for social impact. And this also opens up big perspectives for our healthcare workforce, because you can guess we can make all sorts of PPEs, including the PPE cloth that can become washable and antiviral, allow antiviral protection. Kenda, maybe if you want to add a few words from the industry perspective.
As you say, the mask is the only way, together with the vaccination, to help slow down the spread of the virus. With this Nanotech mask, I think first of all it is eco-friendly because it's reusable and it uses material that are very durable. It is cost-effective, of course, and then it offers a two-way protection, which is not the case for most textile-based masks, which are designed for avoiding spreading droplets around, but offers a low protection in the other way. But this one is really designed to offer two-way protection. It has a high level of protection. You can reach N99, and it is made locally. So there's no reason not to use it. And we hope that with this product, we can help Mauritius and the world fight this COVID-19.
Yes, thank you everybody for having stayed with us. Madhvi, do we have a few minutes, or we are time up?
I think we are quite short on time. I would rather we move to the next presentation.
Okay.
Then we will take all the comments at the end so that we make sure we can finish on time. So thank you, Associate Professor Bao and Mr. Tang, for that most interesting and impressive project. I think Sir Mahmoud Ali and Dr. Robin, please.
Thank you very much, Dr. Madhu. Good afternoon, everyone. So I will be the first one taking you through the presentation. So then Devina is going to take the other half of the presentation. So again, I'm very grateful to the MRC for having funded this project. And for you, this project, you know, the technical word is like the in silico identification of new antiviral pharmacophores from the floral biodiversity. So I know that we have a mixed audience today. So when we talk about in silico here, there are other words like computational or docking studies that we can use. And when we talk about the new antiviral pharmacophores here, is mainly looking for new compounds that can be used as antiviral to manage or to treat the COVID-19. So the team consisted of two local investigators, so myself from the Department of Health Sciences, Faculty of Medicine and Health Sciences, and then Dr. Lobin, who is a postdoctoral fellow from the same department. And we have two additional collaborators, Professor Adriano from Italy and Professor Hassan from Iraq. So just I wanted to throw something next, because people have been talking about, you know, climate change, about SDGs and all these stuff. So I want to have a new thinking also about one aspect that is a key challenge to humankind. And we saw that, we experienced that during the last COVID outbreak. So you will see this was a report, a scheme published by CDC, and it was said that, you know, climate change will introduce new challenges, and with climate change, it is expected that diseases is going to emerge. So this is the question that we have to ask ourselves. If diseases is going to emerge, then the innovation aspect of it, the drug, the management aspect of it has to be catered for. And this is the question that we have to ask ourselves, where our drugs or therapies will come from, because we have seen with the COVID that we don't have approved anti-COVID antiviral drug that can be used to successfully manage this disease. And this is where we have this opportunity of the so-called natural products. When I say natural products here, again, for our audience, we talk about, you know, any plant-based material. It can be animal-based material. It can be any kind of natural medicine that have still standing the test of time. When we talk about natural product, we can also classify and categorize them into marine or terrestrial natural products, which can also be our food, our plants, herbs, spices. And we also have the concept, the so-called zootherapy, that is animal-based products, you know, they are proteins or the derived compound that can be used to manage diseases. So again, coupled with that, you know, with the alarming rate of antibiotic resistance, with the outbreak of re-emerging diseases like Zika, like COVID, like chikungunya. So it's the WHO has called upon for new novel ways of treating these diseases. And you will see that when we talk about traditional medicine or herbal medicine, the main component here is about medicinal plants. When I say medicinal plant, it doesn't mean something that is separate from what we eat as our herbs or spices. So in terms of grouping, it is more than 30,000 species. And the WHO data say that almost more than 80% of the population rely on some kind of natural or holistic medicine, which is termed here as traditional medicine. And one integral component of it is medicinal plants. And interestingly, being part of sub-Saharan Africa, we have more than 40 to 45,000 of higher plant species that have a high potential to be industrially exploited or to derive pharmacophores, that is template, new bioactive compound that can be used to manage existing or emerging diseases. And in terms of genetic resources, it has a great potential to be exploited. So again, natural product research has been propelled and has experienced a renewed interest when we saw the award-winning, the Nobel Prize-winning contribution of Professor Tu Youyou, who discovered the artemisinin from a traditionally used medicinal plant that was being used to manage malaria. And this has propelled again a renewed interest towards natural compound, also called phytochemicals, or sometimes called secondary metabolites. So in terms of herbal market, so it's not only remède de grand-mère. So we are talking here about a global market for herbal medicine, where we see China controlling more than 24% of the global market. India with Moringa oleifera, it has an annual purchase of 1.3 million tons. China export almost 120,000 versus India per year. Europe is a very important importer of raw materials of medicinal plants, and which amounts to more than 1 billion from Africa and Asia. So Africa, including Mauritius, we have been mainly exporter of these raw material that can be used and turned into natural products. So China has more than 40% of the total medicine consumption attributed to indigenous traditional medicine. And now developing countries, including like Japan, they are opting for more traditional or holistic type of treatment rather than pure pharmaceutical products. So in terms of market, so it is expected like to have 1.1 billion by the end of 2023. And the market projected for pharmaceutical derived from natural product is going to experience an increase of 7.2% along 2023. So this is interesting also. Last week, we had a high panel discussion about integrating traditional medicine with African Union, and the WHO representative and the UNESCO also was there. So they have already approved a protocol for the assessment of herbal medicine that want to go into clinical trial. So you see that normally WHO has laid a special section about the development of herb-based products that can be used in our fight against COVID treatment and management. So since the last 10 years, we have been following this small scheme of research about, you know, the documentation first, then moving into the lab-based studies, then moving into the preclinical and bioproduct development. This has been our motto, our flow chart for research in our small team that we have at the Department of Health Sciences. And based on that, from an academic point of view, so we have authored more than 400 publications. Recently, we have secured 2 grants that is industry related, with more than 18 countries as collaborators, and with an impact point more than 1,000 impact factor points. So coming back again to the COVID-19, so the challenge normally is here. So it has already been mentioned. So I was anticipating that an introduction will be done to the COVID. So I'm not going to overemphasize about its structure, but basically one thing that I want to emphasize here is about the structure of the protein that has the binding site with our cells. That is the point of entry and subsequently replication. Here we're talking about a scenario where you have the COVID virus in your body is going to attach, is going to cause the replication. So you will see here normally we have about 3 proteins. In fact, there are multi-target proteins that can be used to do in silico studies and molecular docking to see whether compounds that have already been discovered, that have already been tested for its toxicity, can have a binding effect with the COVID-19 virus. So the challenge, as mentioned earlier, is that the vaccines are available, but the evidence on the long-term efficacy is still debatable. So we don't know how long the immunity is going to last. So the existing approved drugs are already here. So through this project, we have done a drug repurposing property, that is docking studies, to know whether FDA approved drugs or whether drugs that can be extracted from plants or compounds approved can be used against the COVID fight. So basically, this was the rationale of having this study of looking for lead compounds. So for those who are not used to in silico studies, basically we have the target protein, which is going to be on the COVID virus. Then we're going to have a ligand, which is here an inhibitor. The inhibitor can be an existing drug, which is going to be docked using computational studies. And then we identify the hit. So we generate different kind of simulation to get scores. Based on the scores, we do further dynamic studies in order to identify narrow compounds that can be further exploited preclinically or clinical studies. So this procedure is time consuming and can be, it's time effective, cost effective. It's quite interesting to be done remotely. Even during the confinement, we have been able to work on this project. So just to show you, this is the COVID virus, and you will see the structure. There are different kind of binding sites that can be used in the molecular studies, and we focus mainly on the spike glycoprotein. So the objective of the study was divided into 3 main parts. So firstly, we had to construct a database of compounds that were already approved by the FDA, plus did a systematic data mining of the literature to see about African medicinal plants that have antibacterial, antiviral, antimicrobial properties that can be docked against the viral protein. Then the next step that we moved in was using computational studies and in silico molecular docking studies in order to see which compounds or group of compounds can be further exploited. Then the last step, after narrowing, we started to do some quantum and molecular dynamic simulation in order to enlist a couple of those compounds that will move to the in vitro and the preclinical studies. So this is just an overview of the methodology. So the database for the FDA was already available, and it was quite challenging to create our own database for phytochemicals derived from the African medicinal plants and other natural products that was available in the region. So we did the molecular docking and the molecular dynamic studies. So I leave the floor now to Devina, who is going to take a snapshot of the results that we have been able to generate over the last few months. Devina, the floor is yours.
Thank you, Dr. Fauzi. Good afternoon, everyone. I'm hoping that you all can see my screen. I'm not sure about it. Is it working?
Yes, at least I can see it.
Okay. So for the result, we have come up with three main database, which is the FDA approved drug.
Can you put on slideshow, please?
It's not working?
On slideshow, yes.
Yeah. So, and we also came with a plant-derived compound. It was actually getting a compound which has known antiviral properties and has been, registered in natural product database. And we have done extensive data mining by searching database and published articles to find compound which has antiviral, antibacterial, antifungal, anti-Zika from African medicinal plants. So the project we have, as I said, we have divided the project into three. So the part one is about molecular docking of FDA-approved drug against the main coronavirus protease, MPRO. MPRO is very important target because it is vital for the viral life cycle. So to start briefly, to initial screening process, 100 best candidates were selected from a virtual library of 5,000 compounds, which was further screened using accurate manual analysis by superimposition with the entry ligand. And around 30 best hits were then selected on the basis of their binding energy calculation against the MPRO and for further analysis to select the candidate. Then 6 best molecules were subjected to molecular dynamic simulation. Table 1 shows some of the compounds that were subjected to ND simulation with M4, and they are 6 in total, and we have the ligand entry. From all the compounds we are going to describe 3 compounds which have shown significant and stable binding pose with M4. That include Cangula, idarubicin, and sakinavir. Cangula is an anti-platelet drug for intravenous application. Observing the PL and LP contact, Cangula can bind the enzymatic cavity of MPRO, mainly through the N-hydrogen bond with glutamate 166. And according to the RMSF of the protein, does not exceed 2.0 Armstrong. The next compound we have really focused is idarubicin, which is an anthracycline anti-leukemia drug, and the RMSD of idarubicin fits into the MPRO seems to stabilize after 100 nanoseconds, and after the stabilization, all the interactions were enhanced and additional interaction was formed, that is through H bond and hydrophobic contact, as displayed in the figure 3. The interaction was stronger than Cangula and similar to peptide-like inhibitor, the N3. The slide is not working.
Maybe we can try to move it from our side.
Yeah.
I think it's on your side.
So Devina, you can stop sharing screen and then again share it. Okay, it's moving now.
Okay. So we have one of the drugs which is called sakinavir, which is an approved drug by FDA for which is an antiretroviral drug and used together for with other medication in order to treat and prevent HIV and AIDS. Based on the figure 4, we can see that sakinavir has presented the best and most stable pose during the simulation. The PL and NP contact, sakinavir is able to bind with the enzymatic pocket, mainly through H bond with glutamate 166 and glutamine 196 in the agreement with the docking results. Sacchinavia has also exhibited several hydrogen contact with 3 amino acid residues. And we can see that based on the protein and the ligand RMSF graph, they present similar value to Cangula. To conclude the part 1, we can say that based on the overall results, 6 compounds were selected for MD simulation, and we have found that Sacchinavia is able to maintain complexion and hydrogen bond with the main protease, showing satisfactory affinity to MPRO compared to the peptide-like inhibitor N3. And the compound has also displayed a strong potential as therapeutic candidate for COVID-19, and further study are required in order to better understand its biological activities towards this enzymatic target. Then we have the second part, which involves the molecular docking of FDA-approved drug using the LPRO. The LPRO is very essential for the viral replication, and it also performs 2 proteolytic activities that play a significant role in the innate immunity response during the viral infection. Table 2 shows some of the compounds that was selected and subjected to MD simulation based on the docking score. And we can see that we have the ligand as well in the list. And based on the results, we have selected 4 compounds which have shown significant and stable binding pose. This includes ipamidol with and without iodine, zanamivir, panobinostat, and sacchinavir, which has shown stable binding poses. So ipamidol is a drug that is used in the diagnosis of morbidity of head and body. The MD results show that are very good for ipamidol, even though the docking score and the MMGSD output was not remarkable. And the one with which is iodine-free actually bound with the enzymatic pocket of PL-PRO through the pi-pi stacking interaction and several hydrogen bond, and it was found that the iodine-free ipamidol was more stable as compared to its counterpart. The other FDA-approved drug is zanamivir, which is an antiviral drug used to treat infection caused by influenza A and B virus. And observing the PL-RMSD, zanamivir display a discrete binding pose during the simulation time. And this drug has also been found to bind to the enzymatic cavity through several hydrogen bonds, maintaining its complexity and solid interaction with the PL-PRO. Panobinostat is another drug which is another FDA-approved drug, and it is used in the treatment of multiple myeloma. And we have observed that the drug have shown one of the most solid binding poses through the simulation. And also we have observed that there was a stable contact with the enzymatic pocket of the PL-PRO. We have included sacchinavir in the part 2 as well, because we have found that this drug was very effective against the main coronavirus protease, that is MPRO. So for this part with the PL-PRO, the sacchinavir exhibited the acceptable binding pose with PL-PRO, and it was more attractive interaction with PL-PRO enzymatic pocket rather than MPRO, despite of its low docking score. binding to PHYTO through the compound has bind to PHYTO through many hydrogen bond, water-assisted contract, and 2-pi stacking interaction with amino acid residues, as indicated in Figure 10. In conclusion, I will say that among all the tested compound in silico tested compounds, the iodine-free ipaminol and panobinafta exhibited stability similar to the binding pose of crystal ligand, and zanamivir and sakinivir display the lowest docking score and MMG values than ipamidol with no iodine and panobinafta. However, the MD graphic illustrated their potential stability in improved enzymatic cavity. For the first part 3, we have worked with plant-derived compounds, and we had more than 500 compounds in general that we derived from plants, African plants. And as Dr. Fauzi has mentioned, it is quite time-consuming to do data mining, then even to draw the compound on ChemPro and to build a virtual library. So herein we have really concentrated on a batch of 150 structure, and out of these structure we have selected 2 compounds that were subjected to molecular dynamic simulation.
Excuse me, Dr. Levin, you have 3 more minutes to go.
Okay, thank you. So based on the table 3, we can see some of the compounds which show the binding affinity, and retin has one of the highest binding affinities. And those compounds that were selected to molecular dynamic was isoretin, which is a flavonoid T-glycoside. And based on figure 11, we can see that the compound has shown some strong bond with proteins, and also it has a good stability. So another compound that we have selected is aloe, which is an anthraquinone. Aloe is mostly found in aloe species like aloe vera or marron local. And we have found that this compound also exhibits a good interaction and stable bond with the MPRO and PLPRO. As I said, we have gathered many data with respect to plants, actually, that we get in Mauritius over the past month, and we are hereby presenting a case study to illustrate part 3 in general. So we have selected turmeric, which is a widely used medicinal plant in traditional medicine worldwide, and turmeric is also an integral part of our Ayurvedic medicine and our food. So what we really did, we have extracted the essential oil and performed the CMS analysis to elucidate the bioactivity compound. And those compounds which was present in high amount was DOC, again, MPRO and PLPRO. And based on that, the beta sekiphilandrine and alpha zingiberine has showed the highest affinity, again, MPRO and PLPRO. Figure 13 show in general interaction of alpha zingiberine compound with the active site of COVID-19 protein in a 3D representation, and also the figure 13C show the interaction in a 3D scheme. And then we have the beta sekiphilandrine, which has shown the highest binding affinity against PLPRO, and this compound was found to be bind through a number of hydrophobic interactions with several of the amino acid residues. To conclude, I would say that this is a roadmap of the project. And as we all know that COVID-19 is going to stay with us for many years and maybe can turn into a seasonal flu. So it is quite relevant to develop drugs and a natural immune system for prevention purpose. And based on so for what we have obtained as result, we are already in touch with potential collaborators to perform in vitro assays with the best kits. We are also doing a comprehensive assessment of potential plants, especially those ones from Mauritius, so that it can be used and developed into immune booster and nutraceuticals. And for that, we are already negotiating with private partners for nutraceutical development. So this project fully aligned with SDG 3, which is about promoting good health and well-being. We would like to thank the Mauritius Research Council, Research and Innovation Council for supporting this project financially, and also the University of Mauritius. And the team members, actually we are 4 members, and the project was
for the next project. So Mr. Tang and Associate Professor Laximan, please.
Kendall, I will let you do the commercial part. I'll do the 2 technical part, the biomaterial part and the worn out part.
Okay. Yeah, okay. Regarding the price, we have not yet done the final calculation, but we don't expect the mask to be sold for a price exceeding 100 rupees. So I think it is very reasonable. Because we are using only material that are quite standard. And the manufacturing costs, so we have already made, in fact, hundreds of thousands of this kind of mask. So we know on the manufacturing side, we know exactly the cost. The only thing that we still need to calculate is the price for making the nanomembrane.
Mm-hmm. Mm-hmm. And for the biomaterial part and the part on whether it's going to worn out, of course, I won't be able to tell you what is the material that is being used in it. But what I can tell you is that we needed to have, from what Mr. Tang has said before, a proper balance between hydrophobic and hydrophilic. And then we also worked on the mechanical strength. That is why we are sure that this mask can sustain. In fact, the washing that we have done, it's industrial washing. Before the mask has gone for viral testing after washing, these are industrial washing cycles that we have done. And the material that we have optimized also takes into consideration the mechanical strength of the material. And I would also like to add that this nanomembrane is fully recyclable. That is the nanomembrane, that is, if somebody has washed it, let's say 50 times, that person can bring back the nanomembrane to us. We can sterilize it. We can remake the nano fiber. That is, it's just a question of getting back the material, reprocessing it, and making new membrane from it. Therefore, it is a fully recyclable membrane.
We have another question for you, Professor Bao. Just a minute, I'll just read that one.
No. It doesn't have plastic.
Plastic, yeah. All right.
Hits, we constructed the databases, and we're going to do a reverse pharmacology because the compounds come from a plant, from a medicinal plant. So COVID, the curcuma and the COVID example was an example of one of the plant that we have got, because in the interest of time, we didn't give all the plant that we have studied. This is one thing. The next step, normally, we have already a collaborator that's going to test these compounds in vitro for the viral replication. And so this is the next step that we are moving towards. Definitely, after having the in vitro and based on the compound that is already, many of these compounds have already been tested for their toxicity, then the next step will be towards any kind of product development or any kind of clinical study based on the funding that we have secured and the collaborators also. So definitely is getting back to the community, advocating for these plants that are already part of our healthcare system. It's already part of our meal. It's already part of our food system. So just again, advocating for more of these food as a prophylactic to manage or to prevent the viral infection. This is one of the aims of the project also.
Dr. Lubin?
Yeah, sorry. So just to add to what Dr. Palji has mentioned, that we also aim to liaise with the EDB to see what are the already natural products, herbal tea available in the market, and we will try to make the calculation and see if we can test on some of the products in vitro. Because there is a number of herbal tea available in the market already, and some of the plants which they have been using is part of our list.
So in other words, is giving new application to existing herbal remedies based from the COVID, the studies that we have done. So as I mentioned, that many of these already part and parcel of our daily life. So it's just reinventing and re-engineering, the concept of these tisane towards immunoboosters and being used as a dietary supplement or nutraceuticals.
Thank you, Professor. Thank you, Dr. Lubin. I think we have gone through the list of questions. I would like to thank all participants for having taken the time to attend the webinar and to the presenters for the excellent presentations. For those of you who still have some questions and didn't have the opportunity to jot them down, you can send us your queries on the email which is on the screen. So please, if you have comments, questions, queries, things that you've thought about later that you want to ask, do not hesitate, just send us the questions. We'll forward them to the presenters. So on this note, I will thank participants for having made it to the webinar, and thank you again to the presenters. Thank you very much.
Thank you.